Hello students, in today's class we shall study about cytodifferentiation and factors affecting cytodifferentiation. The basic event in development of eukaryotes is the formation of specialized cell, but sometimes unspecialized cell attain maturity to form and acquire special characteristics or function that differ from their original type. This is called as cytodifferentiation. To make it more simpler, we can define cytodifferentiation that occurs either spontaneously or under the stimulus of specific nutritional or hormonal factors. The whole developmental process of plant from callus or explant is termed as cytodifferentiation, or it is also called as cell differentiation. So, if a plant has to be produced from the callus or explant, that process is called as cytodifferentiation, and this requires an important phenomenon to occur, and that phenomenon is your totipotency. Only a cell or a tissue or an organ which has an ability of totipotency can develop into a whole plant. Cytodifferentiation can be divided into three steps. As you can see in this picture, this is 1, 2, and 3. The callus tissue at the time of its initiation and further growth shows a mixed population of small, more rounded, oval, and few elongated cells with dense cytoplasm. With the increase in the subculture number, few cells become elongated with thick wall, and the calli are friable. It is the calli or the calla, singular and plural, what I'm using. By further increasing the subculture number, callus tissue shows maximum xylinogenesis. Xylinogenesis is the differentiation of your parenchyma into secondary wall thickening like xylem, with tracheal elements having a continuous spiral deposition of secondary wall material. If you see in the third picture, where I am pointing, you can clearly see that the tracheal elements having a continuous spiral deposition of secondary wall material. So, these very three pictures shows us how a callus or an explant can develop or undergo a process known as xylinogenesis, or the formation of vascular elements or the vascular tissues. So there are two important factors which support cytodifferentiation. They are the chemical factors and physical factors. First, looking into our chemical factors, we have our phytohormones such as auxin, cytokinin, and gibberellin, which are involved in cytodifferentiation. Auxin at a low concentration stimulates xylinogenesis. This can be proved and explained by an experiment as demonstrated here. Grafting a small vegetative bud on the upper surface of the callus tissue, it has been observed that after few days, the differentiation of vascular tissue in callus tissue takes place below the bud. The observation suggests that the stimulus of vascular tissue differentiation in the callus is provided by the vegetative bud. Now, in the second experiment, if the direct contact between the bud and the callus tissue is broken by placing a semi-permeable membrane at the site of the graft, still the differentiation of vascular tissue occurs. This experiment indicates that the nature of the stimulus is of diffusible chemical. That is, it tells us that whatever the chemicals that is being passed into the callus and which is resulting in the formation of the vascular tissue is diffusible, because though I have placed a semi-permeable membrane, still I was able to observe the vascular tissue formation. This indicates that the chemical which is present in your shoot apex is able to diffuse through their semi-permeable membrane and into your callus to form the vascular tissue. Further, in a third experiment, a piece of callus tissue is placed on a media which do not favor the differentiation of vascular tissue. The callus tissue is totally undifferentiated and is free from vascular elements because it has nothing to trigger the formation of vascular tissue, because it is placed in a media which doesn't have the triggering factor. So the callus tissue is totally undifferentiated and is free from the vascular elements. Now, a V-shaped incision is made on the upper surface of the callus tissue, and the V-shaped cavity is filled with 1% agar block containing auxin and sucrose. The appearance of vascular tissue is again noticed in the callus tissue. But whereas in your control plant, which did not contain the auxin or the sucrose, did not develop any vascular element. These three experiments go on to prove that auxins are very essential for the development of the xylem or differentiation of the xylem or the formation of the vascular tissue. So, next we shall see about the cytokinin. Cytokinin is also another plant hormone. The main importance of cytokinin, as we told earlier, is in the formation of the shoot buds, and also another important, very important function of the cytokinin is the cell division. It has been demonstrated that kinetin added media containing coconut water enhances the tracheal differentiation in the callus tissue of Nicotiana tabacum. Similarly, exogenous cytokines in combination with an auxin do markedly increase the quantity of tracheal elements. That is, it means to tell that cytokines alone or cytokine in combination with auxin can markedly increase the quantity of their plant tracheal elements. Coming to a third hormone, that is gibberellin. Gibberellin interacting with auxin is effective in cell enlargement and tracheal differentiation, but not when used alone. For example, for cultured pea root segment in auxin cytokinin media, the percentage of xylem differentiation is rarely up to 20. in respect to the total cell population. Whereas in the combination of auxin, cytokinin and gibberellin treatments increases the response, giving a higher percentage of differentiated cells compared to the other combination. So this goes on to prove, to tell that the auxins and cytokinins and gibberellin are very important for the formation of the tracheal elements or your vascular tissues. So there are two groups of particularly the regulatory hormones which play an important role, that is auxin and cytokinin. Students, in nature too, auxin and cytokinin act as a biological trigger for wound response. Whenever there is a wound formation in the plant, these two hormones, that is your auxin and cytokinin, act as a biological triggers. Similarly, in the tissue culture laboratory, auxin and cytokines activate the totipotency manifestation in the cell culture. So you can see that in nature also they have an important function to perform. And even in tissue culture laboratories, that is wherein you're growing your plant in the laboratory, they do have an important functions, as they have the function when they are present in the nature. Now, what type of auxins can be used? The most particularly used auxins are indole acetic acid, 2,4-D, or naphthalene acetic acid. And cytokinin, you can use kinetin or 6-benzyl aminopurine. So these favor the in vitro totipotency expression and development of organized cells. Apart from the hormones, even your sugars play an important role. Particularly sucrose in the medium is very essential for cytodifferentiation. Even the effect of auxin on vascular differentiation seems to be closely dependent on the presence of sugar in the medium. At lower level of sucrose, that is 1.5 to 2.5%, only xylem is formed. When a concentration above 4% favors a balance of xylem and phloem. Xylem differentiation to some extent occurs with the incorporation of other carbohydrates such as cellobiose, lactose, raffinose, and glucose at a concentration of 2%. Maltose at a concentration of 2% could partially replace sucrose in xylem and phloem nodule differentiation. It means to tell that most of the sugars, particularly your sucrose, plays an important role. If there is no availability of sucrose, maltose can come to the rescue, because maltose can partially replace the sucrose in the formation of xylem and phloem. Glucose, when it is used alone, causes the development of scattered xylem elements. That is, your xylem and phloem are scattered, as you can see in the monocot plants. Soluble starch at a concentration of 4% stimulates xylem formation. Another hormone, abscisic acid, inhibits cytodifferentiation. On the contrary, the gaseous hormone ethylene production is involved in vascular differentiation. With this, we come to the end of chemical factors. Now let us look into the physical factors. Light. In general, light has proved to be inhibitory in xylinogenesis, although in exceptional cases, as in carrot, it can be a requirement, but it can be replaced by cytokinin. So you can see that the response of light varies depending on the nature of the source of tissue. That is, what type of explant you have chosen, what is the species or genus of the explant you have chosen. Depending on that, the light may be inhibitory or it can have a promotive effect. Temperature. The nature of vascular differentiation is influenced by temperature conditions, whereas high temperature, 35 degrees centigrade, proves stimulatory to xylinogenesis and formation of compact wood as in Jerusalem artichoke. Low temperature causes the development of undifferentiated new tissue. So, a temperature of 35 degrees centigrade is more appreciable for the formation of xylinogenesis. Coming to pressure and water stress. Pressure factors such as increased pressure have been shown to be stimulatory to xylem differentiation. This is probably because of the ethylene production. Because in the previous slide also we have seen that ethylene production stimulates the xylinogenesis. Water stress is also a controlling factor in the initiation of experimentally induced xylinogenesis. So herein you can see a callus is being formed, or this you can consider it as an explant, and B, here you can see the formation of the callus, and this callus in turn, when you provide appropriate nutrients or the hormones, it can develop into shoot. Once the shoot is developed, you can change the hormone wherein you can increase the concentration of the auxin and lower the concentration of the cytokinin to develop the root. So before we could conclude our chapter on cytodifferentiation, I would like to recall you all with what is totipotency is all about. So remember students, whatever we are doing or whatever the manipulation we are doing with a plant, we are doing it in the laboratory, in vitro method. So I can use a explant. You know that explant is a piece or a part of any of the plant, that is the whole plant. I can convert this explant into callus by providing appropriate condition. Or as you saw in organogenesis, I can directly develop the explant into a whole plant. Or by indirected process, I can develop my explant into callus or suspension culture and then develop into a whole plant. Whatever the case may be, you require appropriate nutrients, growth regulators, and appropriate environmental condition to form a whole plant. The formation of the whole plant from the callus or the explant is known as totipotency. So, totipotency is the ability of the cell to regain full genetic potential, to regenerate the tissue or organ, to make a new organ, or organogenesis under appropriate condition. It is commonly seen even in the nature, such as the tuber, the cutting of your rose. All these are like vegetative propagation. Just you sow a tuber and it can grow into a plant. A cutting from a rose, which is a vegetative propagation, can develop into a whole plant. So this is all brought about because of the phenomena known as totipotency. So plants retain the ability to revert to meristematic state even in highly mature and differentiated cells as long as they have viable nucleus and intact membrane. So what we mean to tell is plants retain the ability to revert back to meristematic state even if they are highly mature and differentiated. This can happen only when they have a viable nucleus and an intact membrane. This is what we studied during the differentiation, dedifferentiation, and redifferentiation. But before I could conclude the chapter, I would like to tell that the seed elements, the fiber, xylem, which is having a thick walls, tracheids, these obviously do not divide and exhibit totipotency. These have lost their ability to develop into a new plant, or they've lost their totipotency. So finally, to conclude, I would like to tell that this entire, the physiology or the anatomy can be explored through the phenomena of totipotency under artificial condition, that is under the laboratory condition. With this, we conclude our chapter on cytodifferentiation and factors which influence the cytodifferentiation. Also, we saw the meaning of differentiation, dedifferentiation, and redifferentiation. Apart from this, we studied about organogenesis and the factors which affect the organogenesis. In the next class, we can start with a new chapter. Thank you.